Hi, everybody. I'd like to introduce you to a new service that we have at PATRIC called Taxonomic Classification. To find it, go into Services, click the down arrow under Metagenomics, click on Taxonomic Classification. This is what the page looks like, where you can use either read files directly from your sequencer or assembled contigs from any assembly program and figure out what taxa are in those files. So you may wonder, how does taxonomic classification work? At PATRIC, we use the Wood and Salzburg Kraken program. This is out of Stephen Salzburg's lab. He's probably one of the strongest bioinformatic groups in the country. And generally what we do at PATRIC is, if there's a good program that's publicly available that the community is using and likes, we like to bring it in and wrap it up as a service so that our users can use it. And that's what is the basis of the Taxonomic Classification Service. So let's go back to Taxonomic Classification. You could enter paired reads, single reads, or SRR. If you've uploaded data previously into PATRIC, you could click into the drop box, the down arrow here at the end of the text box, and click on the files that you might want to upload and compare if you wanted to do that. Or you could start typing. If I did SRR, it would go through everything in my workspace to try to find read files that match that, because notice that I'm using reads now. Okay. Single read files, those will also look for read files. But what I want to do for us today is look at the SRA. Often with metagenomic samples, I've been doing this a lot because it's a lot of fun. I go into SRA and look at data, and I can use an accession number. If any of you have downloaded data from SRA or from the European Nucleotide Archive, and it's a metagenomic sample, those things are huge. And the one I did today took an entire hour to download just 1/2 of the read file. So today, we're going to go in and look at, I have 2 here that we can look at the end, but we're going to look at one specific metagenomic file of the infant gut metabiome. And these were taken from this particular one, which is of interest to me. When I went in and looked at the biosample file, these specific ones were taken from preterm infants that were in the hospital and on antibiotics. And we're going to compare it to some that were taken from healthy breastfed infants. So we need to copy and paste this particular SRR number. This is the one we want. This is for the project, that you need this SRR number for the run. And we go into PATRIC and we put it into this box. And then once we've entered that, we've got to move the data to this box, because you could bring in a variety of data sets here. Let's say that you have a metagenomic sample from your sequencing center, and they tell you, oh, you know what, we ran 3 lanes for you in case one went bad, and actually all 3 of them worked. So they're 3 copies of the same data. You could easily load all of those here, and it would make for a more coverage and better data. So you click on this when you want to move things over, and when it's over here, it means we can run that job. So let's look at the parameters here. Right now we only have one algorithm, which is Kraken 2. one database, which is all the genomes. Now here we have two options, save classified sequences. What it's going to do is it's going to map all the reads to all the individual taxa that it can. So if you wanted to, you could have a separate file with the classified sequences. You could also get unclassified sequences. There might be some, some data might not match, and you might want to take it into another resource and try to figure out exactly what that is. So right now I'm going to leave it here at the save. We'll set it as the defaults of no and no, but those options are available to you. There's an output folder for where you need to define to place your data. You can click down and see all the recent ones I've created, or you can start typing test. So I'll put it in my test folder. And generally, I like to have these things have preterm. I like them to have a unique name so I can find them again later. Preterm infant, infant antibiotics. And then when you're ready, you notice now the job will turn, this button will turn blue, and we're ready to submit the job. So let's submit that, and then we get this message saying that we have submitted the job. Sometimes it takes a minute for us to find it on the jobs queue here. But we can click here and see what that looks like. And I can see it's taking a few minutes to catch up here, but eventually it's going to show that job. Oh, here it is now, and it's running already. So this is like a cooking class. I'm going to show you what it looks like when this job has been successfully submitted. So let's look at this one here. And when I highlight this row for taxonomic classification, you notice that there are two icons here that I can use: view the job or report an issue. If you had a job that failed, it's important for us. It's important for you, right? Because you took the time to submit the job. You want to know what's wrong. You can click the report issue button, and it can come back, and you can say, you know, something about the job. Often we get, this job has been queued for more than 24 hours. And, you know, you would think that we would know these things, but PATRIC is a very big resource with a very large database. Often we think everything's going great unless you tell us. So if you have a job that failed, let us know so we can look in and see what the problem is. If you had a job that's been queued for more than 24 hours, let us know so that we can try to devote more resources towards that particular service, especially if it's experiencing heavy usage. But this job completed, so I don't want to report an issue, I want to view it. So let's click on the view icon here. And this tells me specific things about my job, the ID number, how long it took to run, which was 1 hour and 3 minutes. If you click here, it tells you different parameters of the job, and it tells me that I didn't want to do save my unclassified or classified sequences. It also tells me that this SRR number is from a paired read library. I can see right here if you're interested in that. So those are just the boring facts about the job. What's really exciting is the taxonomic report. So let's click on that and let's view it. So this is directly from Kraken, and it's telling me the specifics of this data, which I can download, and we'll look at the interactive chart in a minute. But this is exactly what you need to know about this job. Let me scroll down a bit here so you can see the different column headings. The first column is the percent of fragments covered by the clade rooted at this taxon. So let's look at this Salmonella. 2.63% of all the reads mapped to Salmonella. The second column is the number of fragments covered by the clade rooted at this taxon. So this is the number of reads that comprises this 2.63%. The third one is the number of fragments assigned directly to this taxon. It says 0 here. Well, what does that mean? When you see something like the 0 that's happening here, it's saying that actually the reads are at a lower taxonomic level that they're hitting at. So it's letting you know that 2.63% of the reads are somewhere within Salmonella, but it's not at the genus level, and it's not at the species level. You start to see it here at this particular subspecies, which is in subspecies Enterica. And actually, the majority of all those reads are mapping directly to this particular serovar from this species. So the fourth column is the rank code. So it's telling you that we're at subspecies 2. So unclassified is U, kingdom, phylum, class, order, family, genus, species. Everybody who took biology knows you have some saying that helps you memorize that, so you passed your basic biology class. Okay. Then it gives you the taxon ID and the scientific name. So this table, I've been doing a lot of work with this table for other research. So it gives you, you can download the table, you can download the report, but we also provide this really nice, cute, interactive chart. If we wait a minute here, this chart is drawn by Cronus, and it's showing you how the species are set up across, how the taxa are set up in this particular sample from this infant who is in the hospital. It's a preterm infant in the hospital with on antibiotics. So I'm assuming that it's a child who's not feeling very well. I want to open up a new tab so I can show you the one from the breastfed infant, so the healthy infant. So you can see what it looks like when we compare the two of those together. So I clicked on the Jobs tab, click on this one, view this. I want to see the same folder, view that, and then click here to view the interactive chart. Oh, is this the same one? I opened the same one again. I apologize for that. There's no difference. There we go. Here we go. As we wait. Okay. So this one, it's got a different spectrum. Bifidobacterium, actually that's one of the crucial bacteria that babies have in their guts to give them vitamins and stuff. And actually one of the main components of breast milk is to actually feed these bacteria. So let's say I'm interested in this particular group and I wanted to make a figure about using these. I like this, but I don't care about the proteobacteria. I just care about these guys. So I can click on it and then double click again and it'll reorganize the diagram to show that. And I can even download it at that point to be able to see that. And you can mess around with the size of the font and play with it however you want. So this is kind of fun to play with. Let me go back to the job so that you can see the other data that comes with that job. The chart, you can get the chart independently. You can get the table here. You can get it as a zipped file. This is the full report, or this is just the regular report. The full report has way too much stuff, in my opinion, for anybody to see. That's why we have to wait for it to load. So it goes on across all these taxonomic levels. It's just way too much. I would suggest that you use the shorter one, which is the report text and not the full report text. So today, I showed you how to run the taxonomic classification job using breeds. You can also do it using assembled contigs. We showed you how to find the job and what the results look like. If you have any questions under the Help tab, you can go into Tutorials and you can click down here for Taxonomic Classification, and this will step you through how to submit the job, whether you're doing paired reads, single reads, data directly from the SRA. And at the very bottom of this, which is probably the thing that's most important, pardon me while I scroll quickly down to you all, is I provide the references so that you know when you're writing your paper, you can say, this program, we did it in PATRIC, please always cite us and cite one of our citations to the resource. And also, you can say we use Kraken, the wood, and Salzburg, and here's the paper for it. So thanks, and have a good time using the Taxonomic Classification Service. It's really a lot of fun. Bye.